So, welcome to our two-photon microscope. It's also called Multiphoton. And you want to go here if you have, it could be a live animal or it could be tissue. And if you want to go deep into the tissue, this is where you can do that. So, today I'm fully equipped with this suit and gloves and covering the hair. And I'm pretending I'm doing imaging on this mouse. The way to fit the animals and your sample is something that you really should consider. At this point, it's actually just fitted for slides, and you put your samples onto a slide. You can of course mount living animals and do a lot of custom work on that part. I'll show you in this image, video, how to turn on the system and do the basic operations in the software. First of all, you turn on everything. You can hear already that the laser unit is turned on all the time. So the unit and the chilling device, the cooler for the laser, is running always. The laser is off, and it is controlled in the software. And of course, that's one of the major things, that you don't leave the laser on for when you're not using it. So that is controlled in the software. I will show you in a minute. All the hardware you turn on inside this cabinet. You open the door, click this red button. It says 1. So this guy says 2, so that's a touchpad control unit, and you turn that on. After you've done that, you press 3. That's a computer. And it will take a while, and then you are ready to start up the software. So behind the system, you can see in this camera setup, you can see the laser. Once you will be here, it will be covered with a sight, so everything will be dark inside. But here you see the laser. It's a MiTi deep sea laser that is tunable, and this is a one laser line setup. And in the software, I can show you in a minute how to tune it. In this system, you need manually to align the filters, and they are inserted in this position. So you need to loosen all the screws in order to access the dichroics and the filters. And once opening it, the laser safety is shutting the laser off, so it will not be active. And the first part you see is a beam splitter. So this one is blocking all the high wavelength. This is a setting that we only have this one setting, so you will not remove this one. The other 3 parts you can remove. And I will also show you in the guide. You can see exactly how the dichroics and the filters are set. And also on the table just next to, there's a small booklet of how the beam splitter will split to the different detectors and so on. And all the filters we have currently is in this box. And you should put them in the labeled positions, the ones you're not using. And the way to take out the filters are with this one screwdriver. It's also in the box. And you have different positions here. So up here you loosen by half a turn, and after done that, you can take this filter out, and you can see the dichroics here and the 2 filters. And this should not happen. Someone touched the filter, and you can see a thick mark from a thumb. That will definitely be visible in the image also. You can see this dovetail So that's how you put it in, and you cannot misplace it, but it just takes a few times then you're familiar with that system. So please grip it firmly on this one, because these filters are of course super expensive. Put it in all the way, and then you tighten the screw again. Up here you find the similar screw. Half a turn, taking it out, checking for thumbs. This one is clean. Putting it back in. So these are the filter sets for the 4 detectors. And you see here, this is channel 1, channel 2, and channel 3 and 4. And there are differences. So the 2 first channels are standard PMTs, and the upper ones, the 3 and 4, are GASP PMTs. So these ones are more sensitive than these ones. And you also control this in the software, so you want to put the channels you need the highest signal, you need to put them up in the GASP to get the most out of the system. And for that, you direct the light by the dichroics here, taking out same way. Oh, and I can show you. So this one is putting it away from the light path. So all the light in this case goes directly into channel 2 and 3. 1 and 2. And when it's here, it is splitting to all 4 channels. And there are 3 sets of filter cubes for this part. There's one called a mirror. So that is just reflecting everything up to the GASP detectors. So if you have 2 color imaging, you add in a mirror and you only use the GASPs. If you want to use all 4, then you should do it in the way that it fits you. So this one is a reverse filter. You also see this in the booklet, and that is sending all the light lower than 565 to the GASP detectors. And there's another filter that is reversing or doing it the other way, so sending everything above. So I'll put it back in. And it's important that these settings are also reflected in the software. After this, you put it back on. Without these lids, the system won't work. So this is the position as before, so it is the same position as in the software. All right, so starting up by logging in with the password Olympus. Looks like this, and you map your local drives by your AUID and so on. Then you turn on the software, so the FView 31S software. So that's the main button here. And the system starts out. So you'll be asked if you want to enable the XY stage, and you want to do that. Pressing OK. So the first thing you notice when the software starts up, it's that the laser is off. And that is how it should be, because you need to turn on the laser in the software. I'll show you this in a minute in the software view also. Go to Tools, Configuration, and go to IR Laser Emission, and then you click on for the laser to be active. Still, it says it's you need to turn it on. It's because it takes a few minutes to warm up and to be active. And what you want to do first is anyway to go to Ocular View and use the epifluorescence to locate your sample. Okay, so after activating the Ocular View in the software, you select the filter set that you're interested in, and in my case I'm using GFP. You can now see that you can control the filter cubes in the touchpad control, and you can select the wavelength here. So now this cool LED is on, so that's for the wide field fluorescence, and you can select the wavelength here. So this is a joystick controlling the XY stage, and I'm not going to image the mouse. I'm just having a standard microscope slide, and I put it in here. Moving back here. So secondly, it's very important not to damage the objective. So first you remember that the laser should be controlled, and it's ridiculously expensive, such a laser. Another expensive thing is the objective lens. This is a 25x lens that is motorized correction color, and it's the only lens on the system. It's the stage is controllable, so you can put in whatever animal or specimen you have. Also meaning that the limit is not activated, or is maybe not activated for the slide or whatever you use. So you can take these lever down, so unscrew these ones and take the whole stage down, or you can take it up. But having this flexibility also means that the objective lens can crash into your sample and destroy the sample and itself. So you should really take care that you are going with a coarse focus closely, but not entirely to the specimen. And of course at this point I also need to add some water, because it's a water dipping lens. So doing that, I use my pipette, adding some drops of water to the specimen. So while I look at the objective, I go down with a coarse focus, like this. And I turn on the light by the shutter down here. And I also showed you how to select them from this touchpad here. Of course, this touchpad will be on the outside after the movie. So now I can see my cells, and I use the ocular to get in focus. And after it's in focus, I move to the software, to the observation. The first thing pops up on the screen when you start the computer is this script where you map your drives. Now you see my U drive and O drive. So you can store your data on the data drive and then remove it to one of the network drives when you're done. You can then disconnect manually by clicking this one and remap if another user is going to use it. And the software you need to use is FluView. So this is called FluView and then software. This one is called desktop. So you will be using this one to start the system. And I already started it, so it's active right here. You also have CellSense, so CellSense can handle all the files you acquire. And for that you can refer to another tutorial. I will link to that shortly. You see how CellSense looks, but right now I'll just close it again and focus on the acquisition. And here you see the FluView acquisition layout. You see acquisition here, and you see also the viewer. And then you can actually jump to CellSense if you have it open by clicking this one. You may see straight away that these yellow signs indicate that the laser is not on. So you, in the very top of the software, you see layout. You can here go step by step, basic layout, or some other layouts, and you can also make your own. Start out going up to Tools, Configuration. Then you go to the IR laser and turn it on. Then you'll hear a sound in the laser where it's tuning and it's warming up. Then it goes from off state to unlock state. So in the next few minutes it will warm up and you're not able to use it, but during that time you will mount your sample and install set the correct settings and so on. After turning on the laser, you can go to the filters, and here you see the non-descant filters, and the first I'll introduce you to these in a minute, but just check in the system that these are actually the ones installed. So the first filter is always this barrier filter. Then we have this dichroic and these two filter cubes. So you have to open up the filters and check that these ones actually are in the system. If you need some others, let's say you want a different dichroic, then you go and change to, let's say, the 570. But this is important that you check that these are actually what is installed in the system, because otherwise your experiments will not make any sense. Then you can go to Tool Windows and open this light path. When you do that, it pops up just next to the live window. And you see here a diagram of the microscope. So the reflected non-descant detectors are here. And in this image you see the light goes up, and then the emission goes out to the detectors here. And when you look at this point, non-descant detectors, then you see here how it's organized. So this first filter is a barrier filter, so it's blocking all emission light above 685. This one is a dichroic mirror, and we have three options here. So either a mirror guiding all the light down to the high sensitivity gas detectors, or a filter called 570 directing all the high emission to the PMT detectors, or this reverse filter, which is directing all the high wavelength, the wavelength above 565 down to the gas detectors. When you configure it like this, you send the emission light down to these, and these are actually the filter cubes. So you see here inside each filter cube you have a dichroic mirror, and then you have two bandpass filters. So from 590 to 650 here, and 660 to 740 down here. And in the normal detectors you see a similar cube or another cube, and this cube is what you just saw before in the configuration. So I just go back to the configuration, and then you can see that these are actually the same numbers. Go back to filters. You see here, this is a band path, band filter you see here, and this is a dichroic sending out to the different filter cubes. And you see this is the one we have, and also this 570, and we have also this M for mirror. So these are the three options you have here, and then down to the filter cubes. And please refer to the manual pages next to the microscope, where you see an overview of filter configuration. So nothing changed. This is the setup in the system right now. Then you see, a while ago actually, it was the laser was active, so we can now start selecting these ones. And before I do it, I turn off the light in the room. After you make sure that the microscope is in complete darkness, you can activate these detectors. If you activate them while you have light in the room, they will be oversaturated very fast, and you will hear at some point a warning. But please don't challenge that, because it's not good for the detectors. Right now I have my channels here, and in a minute you will see it with an actual sample. But right now I will just go through the different parameters. And you see, first you see the laser. You can tune it up and down here. And it's a one laser line, so the percentage is the same. Then you have the voltage. You can individually set the voltage for each detector, like this. And similarly with the gain and with the offset. So voltage is the sensitivity. You increase it to get more sensitivity. And then gain, you can enhance the signal. Of course, you also enhance the background. Then offset, that's where you tell the software what is your background, so you can increase and decrease according to how much background you do have. And offset, you have to be careful, so use only very small numbers here. Inside here you have the dye selector, and you allocate the different colors to the different channels. So here channel 1 is blue, and 2 is Elexaflor 488, and so on. And you can add from here, remove and clear. When going through this, first of all, when you have your sample, you will put it into Ocular View. So you press Ocular, and then you can use the epifluorescence, and you can use the normal white light. And if using the epifluorescence, then you select the cube here. So we have RFP or GFP. And then you can close the shutter. And on the table you can, there's a white switch where you can tune the wavelength up and down. And by this way you find your focus, and then you go to LSM mode. So laser scanning mode. And then these are active again. Here we have of course only this one lens, so you can't change that. But you can move it to escape position. So that's always take good care of the distance to your sample. So whenever you have to mount or handle your sample, then move it to escape position, either here or by the set focus control. Because the stage is manual, and you you can change it up and down. You also have to be very cautious with a near limit, the set limit position. So you can, if you put in a mouse, you have to change the limit to another position. Then you have to, if you are imaging a slide. So just make sure that you're not going to crash into the sample or the glass for both the sample and for the microscope's sake. Down here you can touch. You can make the touchpad control dark or bright. So depending on so it's inside with the microscope. But don't. Yeah, you may wonder why it's lighting up in there, but it's for convenience when you are actually handling your sample. You can use that the touchpad control instead of going to the computer. And once you start your scan, it will go into dark mode, so it will not shine light when you image. And then also the LED indicators on the microscope, they are turned off because you don't want light in there when it's not necessary. Here we can activate averaging. So here we have no averaging. You can average by line or by frame, and you can put in a number here. So 16 times averaging is of course reducing the noise a lot. And you can do sequential scan or in line or frame-wise fashion here. And then it pops up with the different groups with the channels, and you can do it like you want to. Here in observation mode, you can make your own configuration. So if you have set up everything, you can save one configuration, and it is easier for you to start up next time. Then we go to Lsm Imaging. Here you can select how you want to scan, with a galvo or with a resonant mode, and you can switch back and forth. And you hear in the galvo, you can hear this tone for the resonant scanner because it's moving very fast. And you want to use resonance scanning when you want to follow movements really fast. And you see the speed is especially when you do line scans, you can scan really fast with a resonance scanner. The time, of course, goes down when you use the galvo scanner, but then you get the best quality. And in resonant, you can do round trip. So you can in galvo scanner you can only do one way, but in resonant you can do both ways, meaning that you are double the speed. Here you can select the area you want to image. You can rotate, and you can zoom in, or you can press 1x and also reset, so go back to original position here. Okay. Then in the right side of the software, you see I can now start the scanning, and I can when I'm done, I can just tell it the series is done. And even though I defined one top and bottom of my sample, I can still go further. So when you set the set position, you should start in the top of your sample and go down, because in that case you can add more stacks when you are deep inside the tissue, and you cannot do that if you are at the top of your sample. So once you are done with a light path, you can either just go to live, You can also close it. Then you have only the live window, and you have the option to go for live, live 2x. Right now I don't have anything in the microscope, so I'll just show you these functions. You can go to a higher speed, so 2x or 4x. So while you're alive, you can go fast through your specimen. And you can see it in tiles, so the separate colors here, or you can see it in a single mode and activate or deactivate however you want. Then you see this high-low, so that's the saturation indicator. So you see here everything is pitch black, so you see blue, and if it's saturated, you see red. And here we see in our left side of the microscope, we did already see the microscope parts here. You can go to information. Here you see everything, how it's acquired. And in this IR laser setting, then you tune the laser. So if I want, let's say I want 920, I'll write it here, I'll use the bar. And you see the laser is on, and it'll take some seconds before it's activated. So now it's activated, and it comes up here, 920 instead. And then you can go back, some 870. And now it's active again. When you have aligned the laser and also the gain and all these things, so you have a good image, you'll go to the rightmost side of the software, and then you can register the top and the start position here and the end position. So you most conveniently you go to the image, and then you press Control, and then you scroll up and down for focusing, and then you go down to a position and press Register, and then you can set the step size according to what you want, how precise you want to acquire, and how much time you would spend doing it. And this was in the normal tab of the series. You can also go to Bright C, and you can put in some positions here in the some set positions, and increase the laser power here. So in this example, we have at this set position we have 5% laser, and down here we have increased to 13. So it will make a gradual increase from 5 to 13 when it's going down through the thickness of the specimen. And that is for channel 1, 2, 3, 4. And you can see the gain, voltage and offset. You can individually change that. So you go here, you can also change the voltage, the gain, offset. So you have many ways to improve the brightness through the set stack. This tab is the auto spherical aberration compensation. That's the correction color around the objective lens. So it's motorized, and you can see here the angle. You can move it. You can't probably hear it, but when you're sitting here, you can hear a faint sound when it's moving around. And you can do this manually by looking at your sample, going through the thickness, and then add positions like this. And you can also do auto adjustment. This next tab is just a viewer. Here you can view your data, and you can also make movie. So when you have some data, you can go into this tab and make movies and turn it around and zoom in and stuff like that. I'll show you that later. First of all, we need to activate the different channels, and in this case, we have green, blue, and red. and the wavelength is set for 940. And for this setting, it's 7% laser power, and the individual voltages you can see here, but that should be tuned for all different lasers. So we're now focusing up in the lymph node using the control and the mouse wheel, and we can see second harmonics in the blue color. We can see the structure of the lymph node to get an idea when we are at the top. And by double-clicking with the mouse, left mouse button, you center the position you click. In the Series tab, you select the top and the bottom of the sample. You can see in the lower right section, you can see the start position is at 12,832. So that's our top position, and then now we are detecting the lower position. And we register that as 13,000. So at this point we have detected the top and the bottom. So register the start position and end position. You can see the positions here. And then for the different channels, we have selected the percentage, 10% of the laser power, and the voltage, the high voltage is set for the individual channels. You can see here. And we're going through the specimen from top to bottom with a step size of 5 microns. And that is something you need to select depending on your sample. And in this, for these lymph nodes, typical experiment would be from the range of 2 microns to maximum 8, depending on the time you want to image. And in order to be sure to capture all cells, we select here 5%, or 5 microns. So we're looking for cells like this in this experiment. And you see another cell popping up here in green. And an experiment like this is 34 set positions, and we're currently at 8. And the scan time is 8 minutes. So moving from series, where we have selected the set, we can go to Bright C, and you can see here the laser percentage and the position. So we have our start position and our end position. And for this sample, we start out using 7% power, and when we go deeper, we of course need more light. And we are then imaging with a 10% laser power. And it will be a linear increase from top to bottom. And also, it's possible to select the high voltage, so you can change also either the laser power and the voltage and the gain, and so on. You can increase the numbers here accordingly. And you see here the channel 1, 2, 3. So now it's almost done, and one thing you may notice is we start in the top of the sample, and that's important you do that, because once we are at this point, we spent 8 minutes, and we could go further, so we could go even deeper by appending some additional series. If we did it the other way, then we couldn't do it. So now I just append one more, because would just check a bit lower position if there are any interesting cells there. And now we're done. And while searching for a new position, we reduced the scan size to 512 by 512 to do it a bit faster. This is the setting from the last position, so I'll just delete this. And we'll register this position. So we are at laser power 8, and this is our current top position. And in addition to increasing the laser, we can here increase the voltage and so on. But we want to go to this auto-spherical aberration compensation and try out first the auto adjustment. So simply click this, and the correction color will change and show you some displays of the different positions. So you can see this correction color angle going from minus something to a higher number. And the automatic adjustment ended up with a correction color position, this minus 33.2 for this one position. And we should do it for a number of positions. And if the automatic is not the optimal, then of course we can do stuff manually also. So we'll try this auto-spherical aberration compensation. And in order to do this, you have to be sure that you activate it in configuration. In configuration, you need to activate the deep focus, and you can do this for different purposes, for the best resolution or depth. If you know you have a very deep sample, then you should take this bar this way. And before I adjust it, I'll increase my resolution to the resolution I will actually use for imaging. And to start out, I can just try the manual first. And here we can see our correction color, and you can manually change it. You can do it stepwise like this. And this is of course dependent on your sample, and you can spend some time really going into details and finding the right position here. I'll just in this case press stop and try out the auto adjustment. So use the auto adjustment, and if you're not happy with it, go to manual. So I'll just press once here, and it will go from the leftmost position up here, and then it would it will jump through some different positions and look for the best intensity. And in this case, it considered the position minus 29.5 as the best position here. And we'll do the same for the different heights. Vil du prøve at køre igennem? So now we just added the end position here, and then we go to bright C and add this during set series. We go from 8 to 12% laser power, and then again we try out our spherical aberration, and we are soon going to image, so I just increase the scan size again and do auto adjustment. In this point we are at we move to the start position, and when we are in the bright C, then we can activate this change during set series and also during manual C. And if we have activated this, we can go move from top to bottom, and now you can see the bottom of our the lowest sample, and we are imaging with a 12% or 11.9, and when we move to the top, the laser is going down to 8%. And the same would of course happen when we image. Now it's done with a spherical operation, and the number is pretty much the same. It's changed a bit in these two sit positions, and you can spend some time doing this manually, and maybe you can increase this and make more points. You can define more points through the sit positions, and fine tune this. And don't forget to apply it to the series. So you can apply it to the set series. And now we image again, and this experiment would take 5 minutes. So while imaging, you can move to the viewer if you don't want to see all these functions while you are looking at your sample. You can go to this View tab and activate only the colors you are looking for. And also we can, just when it's done, we can move to the software called CellSense to analyze the data and to represent it differently. And by clicking up here, you can open CellSense. Now the series is done, and we can look at our data, and we can do it already here. Go to volume and see it like this. And then we can go to the viewer. And if you don't like these white lines called wire cube, then you can deactivate them over here. And we can move around, zoom into our sample. And you can change some settings here and look at it in different ways. Also, you can make movies, so we can add some keyframes like this. Move it and add a new one. Then we can play this small video or sequence, and you can change the numbers by clicking on the keyframe. You can have this division here, the number of, if you want to make it slower, you can add more divisions. So we can see now it's moving very slowly, and this may be too slow. You can also, let's say 15, it's probably too fast. But this is a way to make a small movie. And you save this movie as file here. You can take this file and right-click and click Send to CellSense, and then it's opening in the CellSense where you can do more things. And for this, we have different tutorials for the different systems. When you're done imaging in the configuration, you can go into the first tab is a file location, so you can automatically save files on D drive. You can browse and you can make your own folder here, and of course here you can make some software changes, but please don't do that. If you need to do it, then you need to return to this settings afterwards. This is where you turned on or off the laser. And you can have some fast keys to do stimulation and scan and stop, and these are shown here. Filters, we already did see the non-descan filters, and then the microscope parts here, which will not be changed. So now I'm done imaging and I want to clean the system, power off the laser and shut down the system. So first of all I go to Tools, Configuration, and in the IR laser emission I power the laser off and press OK. And now it shows me that to please turn on the laser. But now I can freely shut down the software and the computer. After the software closes down, you can switch off in the corner here. And after doing that, the power is just behind here. So finally open the cabinet and hit the power button for powering everything off. And clean the system and cover it up.